thank you for the organizers of the meeting for this great opportunity of share my work. So I will start with this cartoon of the integrative genomics approach, in which we try to explain physiopathological states from omics data. But we have to keep in mind that the omics data is just a list of parts, and trying to explain how a cell works from a list of parts, it will be equivalent to try to explain how a Boeing 747 works only from the list of parts. So what we need is an assembly manual, and the assembly manual for the cell, it is the regulatory relationship between all the genes in the cell. So if we have this layer here now in this approach, I would like to say that the system biology field has progressed enormously on reverse engineering these regulatory networks, and we have reached a point in which these networks are accurate enough to being able to predict that changes in the activity of the regulators induce specific gene expression signatures, but also we can use gene expression signatures to infer back which are the regulators that are differentially active. So for this and for going to the layer that is the closest to the gene expression, that is the transcriptional layer, what we can imagine is that if we have two closely related phenotypes, the transition between these two phenotypes can be captured by a specific gene expression signature in which some of the genes are downregulated in the phenotype B versus the phenotype A, while other genes here in red are upregulated, and some other genes are not differentially expressed. So if we can lay over this signature the regulators, we can quickly see that there are some regulators for which the target genes that this regulator repress are mostly enriched among the genes that are downregulated in the signature, and the genes that this regulator activate are more expressed in the signature. While there are other regulators in the other end of the spectrum in which they regulate few genes in the signature, but also some of the regulators some of the regulation relationships are opposite. For example, this regulator activates a gene that is overexpressed, but also repress a gene that is overactive. So we can use this information and the signature to rank all the regulators from the one that is more consistent with the signature to the one that is the less. And this analysis can be done quantitatively, so we can infer the differential activity of these regulators. How we do this in the lab? We first reverse engineer cell context-specific networks from large cell context-specific expression profiles. For that, we use Arachne. And once we have the network and we have a phenotype, we can get the gene expression signatures and then use an algorithm called MARINA for master regulator analysis to infer the differential activity of these regulators. I would like to zoom in this figure. So here, what I'm showing is in the x-axis is the gene expression signature. All the genes were sorted from the one that is more down-regulated to the one that is more up-regulated. And all the bars here represent the target for each of the transcriptional regulators. The red bars here are the genes that are activated by OTX2. The blue ones are the ones that are repressed. And we can see that most of the genes that are activated are enriched among the genes that are down-regulated in the signature. And most of the genes that should be repressed by OTX2 are enriched among the genes that are more expressed in the signature. So with this, we can infer that this gene is less active in the phenotype. This one is in the opposite side of the spectrum. We have most of the targets that are regulated, that are induced by this gene, are enriched among the genes that are overexpressed, and the ones that are repressed are enriched among the genes that are down-regulated. So we can infer that this gene is more active in the phenotype. So if we do this for the full transfactome, I mean for all the transcription factors genome-wide, we can get an activity signature, and if we do this across the full data set, we can infer the activity for each of the regulators in each of the samples. So this process is what we call virtual proteomics. So for developing this algorithm and for validating virtual proteomics, we first work by knocking down transcription factors and then try to infer if we can detect them as differentially active. So for this, we use different transcription factors that we knock down in different cell lines that are part of different tissue types. I'm showing here different subtypes of lymphoma and glioblastoma. And we use the corresponding relevant regulatory networks that we reverse engineer from large collection of expression profiles, again, from B cell and for glioblastoma. So the first method that we tried for inferring the enrichment was just the simple and well-known Fisher-Exot test, in which we can partition the gene expression signature in genes that we detect as differentially expressed and the ones that are not, and we measure the overlap with a set of targets for each transcription factor. So when we did this, we found very good results for a few of the experiments, but not that good for others. In fact, for some were horrible. And the numbers that I'm showing here, sorry I didn't mention before, here the first lane that is rank. The first number is the position in the rank list of transcription factors when they were sorted from the most differentially active, predicted differentially active, to the less predicted differentially active. So we can see that, for example, for MEF2B, our algorithm predicts that when we knock it down, it is in the 34th position. So it's not that good. While for MIB and BCL6, it's very good. It's in the top 10. This number here is the total number of transcription factors that we evaluate. And the number in the middle is the total number of transcription factors that we detect as differentially active. So one problem with this method is that we are not taking into account the mode of action of the TF. I mean, we just check if the regulon is enriched in differentially expressed gene, but we are not testing if the genes that should be activated or repressed are enriched in the opposite side. So we improved a little bit on the Fisher-Exot test, and the modification that we did was separating the signature in up and down-regulated genes, and separating the regulons in targets that are repressed and targets that are activated, just by using correlation between transcription factor and target genes from the big data set that we use to infer the network. By doing this, we saw a small improvement, while in one case, for BCL6 in the cell line, we have even a worse result than before. So what I want to mention about this method is, first of all, the genes that are considered up or down regulated all has the same weight for computing enrichment. But also we have this hard threshold here that is a totally arbitrary threshold that we use to call a gene differential expressed or not. So what we should do is use the full gene expression signature, genome-wide, to do this type of analysis. So for implementing this, we use gene set enrichment analysis. We have an absolute enrichment analysis that is the equivalent to the Fisher-Exot test, and the two tails in which we can test the direction, I mean the mode of action of the transcription factor. When we use these approaches, we find better results for one of the tests, similar results as Fisher-Exot test for the other, and a worse result for this one. So this was really, really disappointing. So what we did was checking a little bit in detail how is the mode of action of the transcription factor and how are we inferring it. So we found that there are some examples in which the expression level of the transcription factor is highly correlated with the target gene, and this is a clear activator. We have very clear repressors, but we have a lot of cases in which, even though there's a very significant mutual information, so a very significant association between the transcription factor and the target, it's very difficult to infer the mode of action. Clearly here, there's a subset of samples in which the transcription factor is an activator, and another subset in which the transcription factor is a repressor. And this could be due to changes in the epigenome between these samples, or maybe changes in the expression level of cofactors. So if we quantify this across the whole network, we can see that this is the distribution of the correlation coefficient between all transcription factor and target genes. We can see that we can fit a mixture of three Gaussians very well to this, in which we have a distribution for the repressors, a distribution for the activator interactions, and a distribution for the interaction of this type that we cannot determine. So we decided to use these two approaches together. I mean, for each target gene, we will use the one tail and the two tail. But then we can mix the results using what we call a three-tail approach, and we weight the contribution for the mixture of the two based on the relative likelihood of each interaction to be a repressor, an activator, or to be undetermined. So when we did this, we got a great improvement in the results. Now all the transcription factors that we knocked down were ranked among the top 10, and also all were inferred to be less active in the phenotype, I mean after the knockdown. So well, we are very happy with this version of the algorithm. We know now that if we knock down a regulator, we can detect it as less active. But what happens if we perturb this regulator without knocking it down? So for doing this, we knock down a serine threonine kinase, STK38, that is a post-translational regulator of MYC. I'm showing here a Western blot result in which, after knocking down STK38, we see that we have a strong decrease in the protein level for STK38 that is parallel by a decrease in the protein level for MYC, but there's no change in the messenger RNA level of MYC measured by qPCR. And this is the estimation of the activity of MYC, and it was very significant. So we moved towards identifying change in activity for regulators from perturbation with small compounds. This is from CMAP dataset, and we use estrogen receptor because it's a nuclear receptor that there are several antagonists in the CMAP dataset. And we can see here that the inference of the activity of estrogen receptor was significantly lower after the perturbation with the antagonists. If we rank all the samples according to the inferred activity for the estrogen receptor, and we overimpose the samples that were treated with the antagonists, we can see that all the samples treated with the antagonists are enriched among the samples that show the lowest activity for the estrogen receptor. But if we do the same with the expression level for the estrogen receptor, we can see that there's no enrichment. So all of these compounds are affecting the activity of estrogen receptor, and we can detect that, while they are not affecting at all the messenger RNA level. So we move then towards a physiologic perturbation in which we use as a model the germinal center reaction. This is the phenotype that is the highest proliferative B cell phenotype in the adult body. And we know that if we perturb these cells with a ligand bind to the B cell receptor, this induces phosphorylation and degradation of BCL6 without inducing any change in its expression. While if we treat these cells with CD40 ligand, there's an increase in the activity of NF-kappa B with increase in expression activity of IRF4 that repress transcriptionally BCL6. So this is the heat map for the expression levels in 6 replicates for stimulating the B cell receptor, the CD40 pathway, or both. These are the bar plot for the same heat map. And these are our inferences from virtual proteomics. And what I want to highlight here is that when we treat the cells with IgM, we see, when we stimulate the B cell receptor, we see no change in BCL6 expression, but we can see a very significant reduction in its activity. And when we treat the cells with CD40, while we see no significant change in NF-kappa B1 and relay, we see a very strong increase in its activity. We have no change, or almost no significant change, in the expression level of IR4, but we see a significant increase in its activity. And of course, we see a decrease in the expression parallel by a decrease in activity of BCL6. So, okay, all of this was done on transcriptional regulator. What happened with the signalome? So we can imagine a regulatory network in which we have transcription factors that regulate targets and signaling genes that regulate the activity of these transcription factors. So if we measure the mutual information between the signaling genes and the targets, we can have an indirect inference of associations between the signaling gene and the target. So we can use this target as a surrogate readout for the activity of the signaling protein. So to validate this, we use, again, the BCR receptor and CD40 experiment. What I'm showing on the left is the expression profile in a heat map way, and what I'm showing on the right is the inferred activity. So we can see that we found 17 from the 29 genes that are part of the BCR pathway as differentially active. The core receptors for the B cell receptor are CD79A and B that are differentially active. And I'm highlighting here that there are some of the genes that are part of the pathway that we can find them as differentially active while there's no change in expression. And this is the same for CD40. The receptor is differentially active, and the NF-kappa B complex, while it's not differentially expressed, we find it as differentially active. So to finalize, what I want to mention is now we can interrogate the regulatory networks with virtual proteomics to transform gene expression signatures into protein activity signatures. And we have validated this at the transfectome level by knocking down transcription factor, knocking down modulators, or using drug perturbation. And we have done this also with physiologic signalings on the signalome. Thank you very much.
Quick question. If instead of RECOMB you were using a regression-based inference method, and then you were using the sign of the edge weights that you get to infer the activators and repressors, how does the performance compare with your method cascaded with the RECOMB?
Well, so we use Spearman correlation to measure the direction. The point is, when we use RECOMB to measure the association between the transcription factor and the target, we use mutual information. So it's we are able to detect non-monotonic associations in that case. But there's a lot of cases, as in this example that I showed before, in which in the data set that we use, we can have subtypes of samples in which the transcription factor in different conditions can act as an activator or as a repressor. In all of these cases, we are not able to infer, here, for example, we are not able to infer properly the regulation mode of the transcription factor on the target. So that's why we use the three-tails approach. We can still infer if there's a significant enrichment among the targets that are differentially active, but we cannot infer, especially for this target, if it is more activated or more repressed.
Thanks very much, Mariano. Is there any way to take advantage of logical conflicts in the data? So for instance, where you have your transcription factor deleted, but you don't observe a response in your data, and so maybe there's a problem either with the regulatory network or there's some compensation in the network itself.
Yeah, this could be in this work we are not evaluating the quality of the network. We're just using the network that we have. In this case, by improving the algorithm, we were able to find all the transcription factors that we knocked down in the top 10. But for sure, if there's not enough variability in the expression profile, especially for the expression level of the transcription factor that you're knocking down, you won't have a good inference of the targets. And the method will fail just because you don't have a good representation in the network for the target of that transcription factor.
Thank you.
A very quick question. In relating the activity of the signaling network to the gene expression profile, the last slides you showed, how do you cope with the timescale difference between the two processes? I mean, signaling changes can happen in tens of minutes, and gene expression changes may happen in hours. So how do you relate these two?
Well, you have to be very careful when you design the experiment. You have to be very careful when you design the experiments. I mean, for the drug perturbations, the experiments were designed in which we did the readout 6 hours after the perturbation. That is enough time, of course, for the drug to act, but also it's enough time to have a change in the expression level. For the knockdown experiments, the readouts were done at 24 or at 48 hours because you have to have enough time to have a change in the protein that you are knocking down to then see the change in the expression level.
Are any of the data sets that you talked about available online if we want to like work with it and like see like how well they match with our previous studies?
Yeah, most of them are online. We can talk offline, and I will tell you which one are already available.
Okay, thank you.